Good afternoon, class. We have a very small class today, but nevertheless, good afternoon. Welcome to this lecture for AGR 2 IPM, Principles and Play, Integrated Pest Management. Today's lecture is Introduction to the Diversity and Ecology of Crop Pests and Diseases, Integrated Pest Management. The aims of this subject are to introduce the diversity of pests and pathogens of crops and the main principles of pest and disease management, primarily in agriculture. But we will touch on an example here and there with agriculture and forestry. Second aim is to introduce the evolutionary history and ecology of pests and pathogens. Now, this second aim here raises two very important questions for consideration. And hopefully we'll be able to answer these questions in more detail by the end of the lecture. The first question, why is it that organisms become pests or pathogens when previously they weren't? Second question for consideration, why do plants suddenly become susceptible to pest and pathogen attack when previously they weren't? Okay. So two important questions for you to think about there. The third aim of the subject is to describe the development of what's known as integrated pest management, and that's something that we'll look at in a lot more detail as we proceed through this subject. So this course or this subject is really a short course in what's called applied agricultural ecology or applied population ecology, which sounds really odd. For those of you that have studied some ecology previously in Years 2, and you might remember we talked about population ecology, community ecology, ecosystems. So essentially, in a nutshell, we're looking at how organisms of the same species interact with each other, how populations of different species interact with each other, and then how all those communities of organisms interact with their abiotic environment. It sounds really odd that we're talking about ecology in this context, within the context of the subject where we're looking at pestilence and plague. But we've come to see that ecology is fundamental to understanding why pests and diseases, pathogen diseases, actually affect plants. So, covering when we cover or when we look at epidemiology of microbial pathogens, that is how these microorganisms cause disease, and we're looking at the population ecology of insects and other animals, we're really studying populations of pests and pathogens and how they interact with populations of crop plants. So you can see already that link between those two groups and the concepts of ecology that we talked about in previous subjects. Let's think of this, consider this title here. Pests and diseases are a major constraint on agricultural productivity around the world. Is that true? Most likely you'll say yes, right? To what extent is agricultural productivity limited? All right, plant pests can cause massive losses of production during outbreaks, disease outbreaks, and during epidemics, but many cause important small losses every year, especially when it comes to pests of roots, plant roots, because these are not immediately obvious. And these losses, or these result in what are called cryptic or hidden losses. Let's consider how, to what extent crop plants are actually affected. Worldwide, the statistics show that pests cause an average of about 40% crop losses, with approximately 1/3 of this due to animals, mainly insect pests, about 1/3 due to microorganisms, more often than not fungi, and about 1/3 due to competition from weeds. And of course, all this depends on the region and on the crop in question. So let's consider this lost productivity according to biogeographical location. Europe and North America face about 30% crop losses, pest disease, 50% in Africa. Let's consider lost productivity according to crop type. 35% loss in wheat, with the major causes being microorganisms and weeds. Rice faces 55% crop loss or lost productivity, with insects being the biggest contributors to those losses. What about losses to weeds? How important are losses to weeds? Losses to weeds are low in crops such as potato, cocoa, grapevines. Here, pathogens, that is microorganisms, tend to be more important. What about losses to insects? Losses to insects are high in the tropics of Asia, Africa, and of course in the Americas, where environmental conditions there favour insect activity all year round. That makes sense. Question?
Yeah.
Yes.
So where does Australia sit?
Where does Australia sit? That depends on the region in Australia. So you can imagine.
North.
Yes, go on.
Yeah, north is going to be more tropical, so insects.
Absolutely.
And then south will be the rest there.
Yes. So in the south, obviously, insects are a problem.
But not as much as in the north.
Not as much in the cooler months. Yep. Cooler months favour wet, tend to favour microorganisms.
Yeah.
Diseases due to microbes. Health and production losses in crops is actually quite complex. Let's think about this. Poor healthy crops can have physical causes, such as poor nutrition, drought, waterlogging, salinity, frost, heat stress. Nika knows about that all too well. Sun scorch, and I was talking about your research project, Nika. Pollution. Or poor health may be due to biological causes. Insect pests, pathogenic microorganisms, and competition from weeds. But more often than not, we have these physical and biological factors interacting in contributing to poor health and reduced productivity of crops. Pests and diseases, what are they? Let's define them a little bit more carefully. Generally, pest, the term pest is defined as any organism that negatively affects crop production. So that means you could theoretically use that term pest for insect pests, microorganisms such as fungi, bacteria, and so on. And indeed, some modern scientists consider all pathogens, that is fungi, bacteria, viruses, they consider them to be microscopic pests. Weeds, small mammals such as rabbits, mice, and rats, and arthropods line insect as pests in a broad ecological sense. So in other words, they consider all those organisms as pests. Although usually microorganisms, that is organisms too small to be seen with the naked eye, are considered to be pathogens, that is the causative agents of diseases. Note that the term pathogen and the disease are two different things, of course. Pathogen is the microorganism that causes disease in the plant, or it might be in an animal.
Yeah.
We've also seen in our previous lecture that many pests and pathogens are considered parasites, in that they obtain their nutrients from another organism, which we refer to as a host. But the term parasite also implies that these organisms cause harm in some way to the host organism. Also another concept of pharmacology. Pests, weeds, and pathogens. And here, of course, we're talking about pests as insect pests and microorganisms we're referring to as pathogens. They share common characteristics. They actually have things in common. You might be thinking, what on earth does a weed have in common with a virus or indeed insect pests, but they actually share common characteristics that enable them to be highly successful as pests in that broad sense of the term pest. The ecology of all these different pest and pathogen organisms in agricultural systems is actually very similar, and pathogens of humans also have the same characteristics. They are, and this is a fundamental principle underlying the whole subject, they all are genetically very variable. Most of them are sexually reproducing, and most of them are outbreeding, that is, they crossbreed. And you can imagine from, or on the basis of genetic background, that this enables them genetically to be highly diverse. And if you have a highly genetically diverse population of any organism, then there's always the chance of at least some of those organisms adapting to changing environments. And they can adapt very, very quickly because of that spread of genetic diversity in the population. And this all goes back to our discussion last year on mending genetics and inheritance and so on. They all, I was going to say they all are, but they all have also a very high reproductive capacity. They reproduce very, very quickly, and they have a high rate of population growth. So you can imagine that this particular characteristic that they share enables their populations to increase dramatically, increase very quickly. So they're very, very good colonisers. They can colonise new areas very quickly. This particular characteristic. As a result of these two common characteristics, pests are all highly adaptive to altered or changing environments, including those environments that are caused or changed by pest control measures. Often pests are able to circumvent pest control measures, especially they can circumvent the use of pesticides and other chemicals. They can circumvent the use of resistant cultivars of plants. So the challenge is therefore to manage the pest pathogens in a sustainable way. So it seems that whatever we throw at them, whether it's chemicals or whether it's resistant or tolerant crop plants, pests and pathogens
They
are
find a
able
way.
to get around. On to the next slide. All pests and diseases adversely affect the productivity of the crop. Many insects simply eat the leaves and other plant parts, but many insects and microbial pathogens have more subtle effects on the plant, like causing mild disease or even just reducing productivity of the crop plants without any obvious symptoms. All the symptoms may in fact be so mild that they're almost impossible to detect. Separation into the categories of pests and pathogens is based largely on size. That is, large organisms that tend to just eat parts of the plants are called pests, and then microscopic ones that interact more intimately with the plants are termed pathogens. But keep in mind, even though I'll use those terms separately, pests and pathogens, the boundaries can be quite blurred. There are many arthropods and other animal pests that are microscopic, just like typical bacteria and fungal microorganisms. So, for example, mites, Phylloxera, nematodes, you can't see them with the naked eye, or often you can't see them with the naked eye. And these are evident in crops only as a result of the damage or disease that they cause. Pathogens like viruses and biotrophic fungi, such as rust and powdery mildews, tend to have a very intimate physiological association with their host. So too do many nematodes and even some of the arthropods, such as aphids or wasps and They tend to have a more a closer symbiotic type of relationship. Even though it's a negative type of relationship, they still have this relationship with the host plant. An example is pictured here to the right. What you're seeing here is a tobacco plant. It's ornamental tobacco, the Kochiana alata, and it's infected with a virus. Have a look at the leaves there. Can you see that mosaic appearance of the leaf? In other words, you have those dark green patches alternating with light green patches. That's a little mosaic type symptom. And this, of course, is tobacco mosaic virus. Tobacco mosaic virus is systemic. It's a systemic virus, so it will travel right through the plant, but it doesn't usually cause the death of the plant. Tobacco plants will still continue to grow, they will flower, they will set seed. So even though the health is somewhat affected, the health of the plant is somewhat affected, it doesn't mean they die. So you can imagine that this type of relationship between the virus and the plant is quite a subtle one. So we normally only pick it because of that characteristic mosaic system of the leaves. Let's revise some important terms from last week. We can classify pathogens as biotrophic pathogens. This means that the pathogen is one that obtains its nutrients from living host cells. And a very well adapted biotrophic pathogen or parasite will tend to obtain nutrients from its host without killing it. And these are often the most successful ones. They don't kill their host plant, which means that they can continue to live off that plant or within that plant and just continue to reproduce. A necrotrophic pathogen or necrotrophic parasite rapidly kills the host tissue. So these are the ones that will produce really potent toxins and enzymes that can degrade plant cell walls. And this type of pathogen will live off the dead tissue as a saprotroph. This is a new term for you. This is a hemibiotropic plant, which has that typical biotrophic phase where it lives off living host cells, but eventually it does kill the host tissue on which it reproduces. So this is somewhere in between that biotroph and necrotroph. And here we have a couple of examples of what's meant by an organism that has that more subtle relationship with the plant and one that simply eats it. The eucalypt leaf and the leaf spots that you can see on this leaf is a disease caused by microscopic fungus. And to the bottom of this image, we can see very clearly damage caused by a chewing insect. Which is eating part of the leaf and moved on. So a much simpler relationship there between the pest and the plant. Let's think about this term briefly, integrated pest management. And we certainly won't go into detail here. Save this for later lectures. Integrated pest management considers the management of pests and diseases using ecological principles. This includes organisms that have a negative effect on crops and pests, and it adds and emphasises another dimension to the word integration, which is implied in the integrated pest management or IPM to be discussed later. So originally that word integrated in IPM referred only to the integration of various control measures to be used against specific pests. But now it's used also to refer to the integration of control measures against all the organisms that are adversely affecting a particular crop. That is all the pests and pathogens that are on a specific crop at a specific point in time. So this redefinition, if you like, that word integrated ties in with the whole crop and whole farm concept of management, that is integrated crop management. For example, the amount of damage caused by pests can be exacerbated by the poor nutrition of the crop, and nutrition has to be considered. It can be exacerbated by poor soil management or poor management practices in the world. Okay, now we're going to try to answer those two questions that I posed to you early on. What causes a pest or microorganism become a pest or microorganism when it previously wasn't? And what causes plants to suddenly become susceptible when they weren't previously susceptible to pests and pathogens? To answer these questions, we need to look at the ecological origin of pest and disease problems. Crop pests usually result from changes in the ecological interactions of a crop species. Changes in the ecological interactions of a crop species. That is, changes in those interactions compared to the interactions that originally evolved in what we call the centre of origin of the crop. So if we consider that term for just a moment, I'll come back to it. The centre of origin of the crop. That is where that crop plant originally from. Where did it originate from biogeographically? Wherever that crop first originated from, it interacted with its environment. In other words, it had a particular ecology that suited the plant to all the interactions with other organisms in the environment and also the abiotic environment. So we can think about that particular plant as having a particular centre of origin, a particular ecology. If we change that ecology, that will affect the ecology, that will affect interactions of that plant. Okay? Yes.
Just a question, like, is it like when you say, when a plant originates,
Yes.
say a fruit tree, so the aim for that fruit tree is to reproduce.
Absolutely.
So what we call
repeat what you're saying.
What we call pathogens,
Yes.
well, for the plant, actually, probably they're not pathogens because, well, that's part of their life cycle.
That's right, exactly right. And this is what this is talking about. When we say the ecology of, for example, a fruit tree.
Yeah, no, I'm just understanding.
Yeah, for example, that fruit tree has evolved with other organisms. Yeah? It's co-evolved with other organisms. So those, in order for that fruit tree to keep reproducing, it must tolerate the other organisms within its ecosystem. So correct. So those organisms in that place of origin are actually not pathogens and pests. Or certainly not major pathogens and pests. They certainly don't prevent that fruit tree from reproducing. If it did, that fruit tree would go from that region. Absolutely correct. Yeah. Anyway, I'll come back to that term centre of origin. Also, crop pests and disease problems originate because of changes in the crop species itself. And I'll come back to that also. All right, so we're going to look at a few different factors here which causes pest or has contributed in some way to our current pest and disease problems. Pest and disease problems occur due to movement of organisms to new biological regions, to new bioregions. In other words, we can refer to them, to these organisms, as new encounter pests and diseases or new environment weeds. Let's look at how this can occur. This can occur due to movement of organisms between continents. For example, downy mildew and phylloxera of grapevines moved from North America to Europe. wheat, striped rice, and blackberry moved from Western Europe to Australia, or we can have movement within regions. So, for example, cotton boll weevil moved from Mexico in Central America to the USA. Became an enormous problem. Phytophthora cinnamoni moved from our tropics in the north down to southern Australia. Another enormous problem because of movement of organisms to new regions. This can also occur due to movement of crops to new countries where they encounter new pests and they haven't evolved with those pests in their original centre of origin. Cocoa in West Africa, Papua New Guinea and Indonesia became very, very susceptible to diseases such as cocoa swollen tooth virus, vascular streak dieback of cocoa and cocoa pod borer for this very reason. Let's talk about these centres of origin. Once again, centres of origin are also referred to as centres of crop diversity, which you can see at the top of the screen there. They're named after a chap called Vavilov. He was a Russian agriculturalist, and I'll come back to him in just a moment. So sometimes these centres of origin of important crop names are even referred to as Vavilov centres, named after Vavilov. This is fundamental knowledge for agriculture. Every crop actually has a fascinating history, and this can be really important knowledge for current management of crops, particularly for management of pests and diseases. And I'll show you how they've returned to centres of origin to find answers to current pest and disease problems. So you can see here some of these centres of origin. Starting here on the left, can you see my cursor there on the screen?
No. Oh, yes, yes.
Okay, so there we go. So the central region of America. This is one of the centres of origin, and you can see what crop plants were thought to have originated in this region. Avocado, cocoa, corn, bean, squash. And remember, we might not be talking about our current highly modified crops, but the ancestors of today's crop plants originated in these centres of origin. Let's look at the northern parts of South America. So places like Ecuador, Peru, Bolivia. This is an important centre of origin for pineapple, cassava, potato, and tomato. Africa, another important centre of origin for, just some examples are shown here by the way, sorghum and coffee. Then we go up to Europe, the Mediterranean regions and the Middle East, we have melon, grape, cherry, apple, plum, wheat, barley. And then over to the Indo-Asian region, we have tea, rice, orange, and bananas. Okay? So you can look into these more if you're interested. Really fascinating reading.
So how do they get there? Like, how do they get this sort of?
Okay, I'll come to that in just a minute. How do they actually find out where these plants originated from? All right, it all goes back to the Russian agriculturalist Nikolai Vavilov. Vavilov was very, very passionate about agriculture, but he was also fascinated by the origin of plants. And he had a vision. He travelled around the world with many of his colleagues, and he searched for and collected germplasm, seeds, and any plant tissue he could from around the world. And he had this vision. He wanted to start this national breeding program in Russia. And here he is pictured here at a market in
Mexico.
Mexico, thank you, buying bean seeds. So all the research that he did with his colleagues, and he set up hundreds of research centres, all the research he did led him to believe that there were major centres of origin of crop plants around the world. And he provided a lot of evidence for this, and this was later modified to include also secondary centres of origin as more and more information became available. Vavilov was eventually imprisoned and killed by Stalin in 1943 because of his scientific views. The very same views taught in this subject and which underpin modern agricultural science. If you're interested on learning more about how they worked out their centres of origin and secondary centres of origin, here are two of his rewritten publications, which you can actually obtain from the library at La Trobe University. Okay, let's consider the ecological origin of pest problems, and we're still on this first point here. Pest problems and disease problems arise due to movement of organisms to new bioregions. And remember, we can refer to them as new encounter pests and diseases or new environment. This can involve movement of plants or animals from one bioregion to a similar climatic zone, where unhindered by their normal pests and parasites which are left behind, they become weeds or pests themselves. And this is referred to as the enemy release hypothesis. That is, you have an organism that becomes a pest, pathogen, because it's left its enemies behind. It's released its own enemies, which normally keep it under control. For example, a plant such as blackberry or prickly pear, or an animal such as a rabbit, cane toad, snail, slug, is taken to a new bioregion where it escapes from its normal enemies that it had in its original bioregion. So remember that as the enemy release hypothesis. Biological control measures usually involve bringing that co-evolved enemy that's left behind to the new bioregion to try to control what has now become a pest. Of course, this should occur after much testing. Make sure that the biocontrol agent doesn't itself become a pest in the new bioregion. And this has been especially important in Australia, which has been the most isolated bioregion in the world. Still on the same point here, movement of insects from one bioregion to a similar climatic zone where unencumbered by co-evolved pests and parasites or encountering specific environments, they become pests. So here's another example listed here. Cottony cushion scale moved from Australia to California. Cottony cushion scale is pictured here. This is a scale pest of citrus, and it was introduced from Australia, but it probably originated in Asia, to California, where it became an enormous destructive pest. It had to be controlled by biological control methods. That is, they took from Australia the original predator, the Vidalia beetle, which looks much like a ladybird, or a type of ladybird, and that ended up controlling the scale over in California. Ecological origin of this problem. Factor number 2. Problems may arise, problems may arise due to changes in the way that we grow crops. Okay? This is what we normally think of when we think about why pest pathogen problems occur. How have we changed the way that we grow crops? Greatly increased use of monocultures. That's normally an obvious one. Reduced or altered crop rotations. Another one that everyone's quite aware of. Changed tillage practices. This is a change in the way that we grow crops that we're actually not really understanding how it's affecting or how it's increasing our problems with certain pests and pathogens. Because we're really in a bit of a bind here. We're being told that no-till or minimal till is the way to go on one hand. On the other hand, more evidence is presenting itself that this is one way that we're contributing to these problems. Not an easy problem to fix. Altered plant spacing and increased use of chemical pesticides. So there's different ways that we're growing crops now. We're reducing the genetic diversity of our crop populations. That is, we moved away from mixed native vegetation and mixed traditional cropping to monoculture. It's the essence of industrial agriculture. More crowding together of plants, less genetic diversity within crop species. Why do we want less genetic diversity within crop species? Because we want
the stable product, like
exactly right. We want them to germinate at the same time. We want them to grow at the same rate. We want them to flower at the same time and produce their products at the same time. To achieve that, we need less genetic diversity. That means we need increased homozygosity, and therefore we have far less variation, genetic variation. We also have more extensive contiguous areas of particular crop species, and so all these taken together mean that our monocultures, parasites of those pest species, and those pest species therefore will expand in terms of numbers and so become even greater as their numbers increase. During the process of domestication, also many of our crop species changed. They've changed from outbreeding wild species to inbreeding crop species. This is another way that we reduce genetic diversity. In conclusion, we have greatly reduced the genetic diversity and adaptability of crop species. So keep in mind, once we reduce genetic diversity, we reduce the ability of the crop plants to adapt. We reduce their ability to evolve, while the pests maintain their ability to evolve and to adapt to changing environments. In other words, we have created most of our own pest and disease problems.
was it done intentionally? Or?
No, not intentionally. We did not do that intentionally.
No, I know that. No, I'm saying like this.
Yeah, no, we did these things normally to improve our processes, to improve to improve efficiency of farming practices, increase profits, and so on. We definitely don't do these things with the aim of increasing pest and disease problems.
That's right. Yeah.
It was just unfortunately an effect, a secondary effect of other measures that we will put into place. All right, genetic diversity. How important is genetic diversity? It's incredibly important. Genetic diversity is a feature of natural vegetation. Here's just one example of rainforest in Indonesia. It could be one of our own rainforests in Australia. There are more than 200 tree species in a 10-hectare area. This doesn't even come close to the number of species of other plants in that same area. Already you can start to think about it in this way. Why doesn't this natural vegetation, why does it never succumb to epidemics or pandemics of pests and disease? Why doesn't it? Whereas you could have a monoculture nearby that is decimated. Genetic diversity is an important key. Here we have conversion of that incredibly diverse rainforest to a rice monoculture. Genetic diversity, once again, this is a feature though of traditional agriculture. This is old traditional agriculture. In this same area we have banana, cassava and taro. You can see some mild disease there. At the bottom of the screen, you can see a mild case of taro blight caused by the fungus Phytophthora colocasia. Okay. Keep that in mind. It's only a mild case. Everything else looks relatively healthy. Even that taro plant looks relatively healthy. Now look at what happens to taro when we try on monoculture of taro. We have a catastrophic epidemic of taro blight. So what we originally had as a very, very mild case of disease has now become an epidemic and destroyed basically the entire crop. Once again, genetic diversity is key. This brings us to the third factor, which can explain the origin of the pest and disease problems. Vegetative propagation of crops. Once again, we try to keep everything the same. We want everything to grow at the same rate. We want everything to flower at the same rate, and so on. We also want to increase profits, of course. So vegetative propagation of crops can increase problems with viruses which are transmitted in that propagated plant tissue. For example, cuttings, using cuttings to start off new populations of that plant. It's also, vegetative propagation also results in extreme uniformity of plant populations in the crop. Once again, reduced genetic diversity. For example, grapevines, bananas, sugarcane, potatoes, cocoa, and all fruits. Factor number 4, pest and disease problems occur because we've reduced the resistance of the crop plants. We've actually altered the way that crop plants actually resist or tolerate certain organisms, attacking certain organisms. We've changed the plants, we've changed their defences, their defence mechanisms. So, for example, we select the plants that are highly colourful. Okay, so which makes sense, but in doing so over time and over repeated generations, we actually remove the bitter tasting defence compounds like phenols and alkaloids that we learned about in immune systems too. We remove these compounds during our traditional plant breeding methods. In other words, we increase the susceptibility of those crop plants to pest attack, to disease problems. We're removing again the defence mechanisms. Another way that we do this, that is we change or alter those crop plants, is that we select only for high yield. We neglect to select for resistance or tolerance. That is resistance or tolerance to pests, resistance or tolerance to disease-causing pathogens, resistance or tolerance to environmental stress, frost, heat stress, so on. Once again, you come back to Luca's research project. Sorry, Luca.
That's all right.
The opportunity is presented. And of course now, as you probably know, they do try to select varieties that have tolerance or resistance to a certain pathogen? Is that enough to have resistance to one pathogen? What about that integrated crop management approach? Just another one to consider. All right, let's move on now and visit the range of crop pests. Firstly, weeds, all other plants that obviously compete with your crop plants. Mostly these competitors crop species, and they often co-evolve with those crops, or they've been introduced from a similar climatic zone. And obviously they compete with your crop plants for livelihoods and expense. Australia has been very prone to invasion by weeds from similar climatic zones in Africa, North and South America, and Mediterranean. Here's our classic example of invasive weed, prickly pear, this cactus species, introduced to Australia from South America in about 1830. And by 1925, it had expanded to ruin 24 million hectares of woodland grazing in northern New South Wales and southern Queensland, as you can see by this photo below. I must say the fruit is very tasty eating. Has anyone tasted it?
Oh yeah, of course.
Very good. If you can get past those horrible spines.
Just put in water. You put them in water, and then you just scrape it.
Oh,
Like don't touch it with your hands,
no.
because otherwise you're never gonna get rid of it. But, yeah.
All right. So, what was the outcome of this invasion? Entomologists searched in the centre of origin. Here's where you use those Babcock centres of origin. They searched in the centre of origin of prickly pear for its co-evolved enemies. Many were tested, and this led in 1925 to the release of Cactoblastis moth, one of its co-evolved pests, which caused the population of prickly pear to crash by 1932. And the population of prickly pear has fluctuated and a low level since, which is very important in terms of biological control, because we need enough prickly pear remaining to maintain a low population of Cactoblastis. One of the important considerations. Here we have photos showing before Cactoblastis was introduced and after Cactoblastis. Very impressive example of biological control. Okay, the range of crop pests. Number 2, small animals, slugs, snails, mammals, birds. Obviously some of them are not so small. Slugs and snails, the problem species are mainly introduced species, these important pests, pustules, rabbits, foxes, cats, mice, rats, and so on. And of course the classic case of an animal pest is a rabbit. Classic pest in Australia, introduced from a not so similar or similar climate in England, but they soon adapted. Highly adaptable. This goes back to those common characteristics that pest species have. Yeah. Highly adaptable, and they reproduce very, very quickly. So when the rabbits were introduced, they were released from the normal parasites and predators that normally keep their numbers under control. They filled an open niche in Australia, multiplied plate proportions, spread over 2/3 of the continent. Note their adaptability, as you see. And everyone's familiar with the viruses, myxomatosis, caliciviruses, which were introduced. But of course, what happens when you have a highly adaptable species?
They adapt to.
They adapt to anything, including the release of viruses. So obviously that's not the way to control them.
They put out a new one.
Sorry?
They put out a new
They
strain
did.
of Calicia.
They did, yes.
Just keep doing it.
What do you
over and over
predict?
again.
What do you predict?
Well, population will
Other
decrease,
What that does, when you
decrease, and then
expose-
goes up again.
When you expose any highly adaptable organism with high reproductive capacity, high genetic diversity. When you expose that
Resistance.
population, you're going to select for the resistant individuals. So like I said, the population will drop and then it will jump up again. You're facing the same problem. Okay, this map just shows the spread of the European wild rabbit in Australia. You can see 1817, the rabbit was first released, farming farm interesting, and you can see it's spread right up
Winters.
to 1980. And still spreading.
See, they covered quite a big area, didn't they?
Amazing. Absolutely amazing.
Like from Geelong up to
Yeah.
almost past Alice Springs.
Yeah,
Ten and
right.
three.
Those northern, way past, right up to those northern states. That's incredible. But wait until you see one of our other pets. I think on the next slide. I'll give you a minute to look at that because that's quite interesting.
The next slide, that's Brumbies, huh?
Next slide.
Horses.
All right. Here we have just some maps here showing the spread of other pest species.
Ah,
The first one,
cats.
cats. Look at that spread there.
Can't stay in
Cats. How highly successful are they as a pest species?
there.
Okay, feral cats. To the right of that, we have donkeys. Second row, we have foxes. Said to be more limited, their spread compared to the cats. We have goats. To the right, bottom left, pigs, and bottom right, rabbits.
I'm surprised camels didn't make it on them.
Yes, yeah. I think camel populations probably not up there
I
yet.
thought they were about up there with about donkeys.
Oh, did you? Oh, maybe. Yeah,
Yeah.
actually, you might be right.
Look at foxes, they're killing all the small marsupials and stuff like that. They've been like a series.
Probably not as much as the cats are.
Not as much as the cats. I think the cats are clear winners.
Yeah.
Yeah. Okay.
Well, this says where they spread, is it?
Yeah, just to show you.
Not the
spread.
population, actually, is
No,
it?
just showing you
So that's
spread.
one, yeah.
Yeah, no, I've read that the cats kill more native species than any other.
Than any other, feral animal.
Yeah.
I've heard that too. Haven't seen it studied, but it makes a lot of sense.
It's pretty hard to monitor
It is.
on that kind of scale.
Yeah, yeah. But knowing how successful they are, how effective they are.
Yeah.
As hunters, it does actually make sense. All right, on the next slide, we have our third group of pests, arthropods, which you're very familiar with now, after Mackenzie's component of the subject. And this slide just shows the copepod flora, which is a huge problem in cocoa production overseas. Caused by this tiny rock that you can see to the bottom left of the screen. And the grub off to the right on the line. Okay, group of pests, number 4. Nematodes. There are more species of nematodes than insects. That's amazing. And they're more diverse than viruses and bacteria. And probably more important, but they're underestimated. Of course, they live mainly in soil and they attack roots, causing those hidden or cryptic losses. The organisms are just visible sometimes as tiny roundworms, but they're very difficult to detect in soil. Range of crop pests, group number 5, there's the fungi and fungus-like organisms. Along with insects, they tend to be the main pests of crop plants because of their dispersal, their ability to penetrate into plant tissues and dissolve cellulose and plant cell walls. The organisms are often visible as signs on the disease parts of the plants. And you can see some examples here, leaf spot on eucalypt, left. Bottom left of the screen, our favourite penicillium fungus growing on citrus, and to the right, powdery mildew on cucumber leaves.
That's the one.
Yes, but that's okay. Go on.
Just wanted to double check that this
Oh, right.
and it
Yeah.
isn't working. Sorry to interrupt.
Yeah. Apparently it is working, but I'm not using it.
Can I just press it?
Sure.
quickly?
Yes, can I continue with that? Thank you. You'll just be on my recording. That's all right. Okay, many fungi form those biotropic relationships that we were talking about. Those biotropic symbiosis with the host plants. And this is a point of difference, as we said earlier, from many insects that simply eat the plants rather than forming those physiological relationships. This is an important one here, top right screen, wheat striker, caused by the fungus, Puccinia triformis. To the left of the screen, bottom left of the screen, you can see some mesophyll cells. There's from the plant leaves. And you can also just make out that there are very small depressions in between the cells. These are the hyphae of the rust fungus growing in between the cells. You can also see these depressions within the cells, these small circular depressions. These are called claustoria. And claustoria are basically the penetrative structures that the fungal hyphae produce. They insert into the actual cells and you see the fungus causes symptoms. Range of crop pests, number 6, bacteria. A few of the genera are very important as plant pathogens, for example, five-blighted pears, but bacteria generally lack that penetrating ability. Apart from a very special case, Agrobacterium tumefaciens, which we learned about in Living Systems 2, biotechnology component. Obviously, some of you haven't gotten to that stage yet. Bacterial pathogens may be visible en masse as or in the form of slimy ooze that comes from the disease. And you can see an example here, bacterial root of banana, otherwise known as milkweed. Range of crop pests, number 7, viruses. Everyone remembers from Living Systems last year that viruses are usually just bits of a nucleic acid surrounded by protein protein. We say usually bits of RNA when it comes to plant viruses, but the nucleic acid could also be bits of DNA enclosed by that capsid. Viruses are normally transferred or vectored from one plant to another by fungal organisms. They don't have their own ability to be transmitted from one plant to the next. These vectors are normally insect vectors, for example, aphids. And the study of viruses actually initiated, or in part initiated, the science of molecular biology. My favourite scientific discipline. The origin of viruses are unknown. There's only theories at the moment. You can always read up on that. But the possibility of new virus diseases emerging is actually quite high. So it's a serious threat to agriculture. Viruses, of course, are so small that they're not visible except under a high-powered electron microscope. And once again, our classic symptom of virus is that mosaic symptom. So to the left and right in this photo. Now we're going to look at some examples of catastrophic threatening diseases. But catastrophic not just in terms of agricultural loss, but in the way that they affected people's lives. And also how they affect the way that we think about pests and diseases in agriculture and think about how to manage them, what's causing them, and how to manage them. Okay, but before we do, would anyone like a quick break?
Yep. Sold.
I will just mute. Put us all on mute for 10 minutes or so.
All right, we're going to start with this classic example of late blight of potato in Ireland, which I'm sure you're all familiar with. It was caused by the fungus Phytophthora infestans. And the Irish potato famine was actually caused by exploitative social and political conditions, not just the devastating epidemics of the late blight disease that occurred repeatedly in Ireland in the mid-1840s. So by way of introduction, let's just talk about the potato. Solanum, I should say, tuberosum, was first introduced to Europe in the early 1500s, following Columbus's voyages to Central America, where we find the centre of origin for potato. It grew extremely well in the absence of co-evolved pests and diseases. That is, it wasn't seriously damaged by local organisms that it evolved with. It became the staple diet of poorer farmers in Europe and Ireland, and it ended up being grown as an extensive monoculture. What we had here was an example of enemy release. So remember that enemy release hypothesis? The crop probably lost resistance to diseases in the absence of selective pressure against the disease for a period which spanned about 300 years, that is from 1510 to 1845. And when we talk about selective pressure, we're really talking about that process of natural selection. And for those of you who haven't finished Living Systems 2, it's something that we'll cover in more detail in one of those later lectures. Evolution by means of natural selection. In the 1840s, Phytophthora infestans found its way to Europe, where it met a crowded crop, a uniform crop, and a susceptible crop, which had lost that resistance. So the potato crops were largely destroyed in Ireland several years in succession, 1845 to 1847. Over 1 million native Irish died and 2 million emigrated to places like Australia and the USA. Potato late blight, the disease, what does it look like? Here we have a healthy potato plant, top left of screen, and just to the right, this potato plant's obviously been destroyed by the late blight disease. Here are some infected potato tubers to the right, and you can see that the Phytophthora tends to be concentrated towards the outside of the fruit. But to the bottom left of screen, we can see this advanced rotting of the potato tuber by Phytophthora. This image on the bottom right of screen shows or depicts a poor Irish family digging for potato tubers to avoid starvation. Okay, let's look at the spread of potato late blight. In this map to the left of screen, mid-July in 1845 is depicted by this dark brown region. So let's look at that. Around the area of Belgium is where it was first detected, where late blight was first detected. Then you can see the spread over a period of just months. So in light brown, we can see the spread of late blight from July to mid-August. Then we can see that region becoming increasingly widespread by mid-September, and then by mid-October of the same year, the yellow regions. So we can see that it's reached Spain, Italy, up through the Scandinavian countries, and of course the entire region of Ireland was infected with late blight. Okay, just to the right of that, we can see one of the late blight lesions, early lesions that develops on the leaf. More infected potato tubers to the right in A and B, And in C, we can see the spores of Phytophthora. Mhm. That's correct. Okay, so how important was potato late blight? Number one, combined with the social injustices, potato late blight had tremendous social consequences in Ireland, which are actually still being felt today. Yeah. Anyone heard of the Troubles? The Troubles, this is the conflict, even referred to as a civil war, that was raging up until recently in Ireland, Northern Ireland versus the rest of Ireland. This actually shows that the nature of famines often have a social and political dimension. So there's lots of literature out there on these consequences of potato late blight, if you'd like to read further on that. Okay, factor number two, the importance of potato late blight led to the first demonstration that a microbe could cause disease when it invades a healthy organism. Before then, fungi were simply regarded as organisms that lived on dead organic matter. That is, they were just regarded as saprotrophs. It wasn't recognised that microorganisms cause disease. So this heralded the beginning of what was referred to as the germ theory of disease, that germs or microorganisms can cause disease. This occurred in the 1800s, and we still adhere to this germ theory of disease today in both agriculture and medicine. The third important consequence of potato late blight. This heralded the first signs of understanding of the biogeographic, ecological, and evolutionary aspects of disease in crops. This is when people started to recognise what was causing epidemics of pests and disease in agricultural crops. In other words, one, the movement of organisms between continents, that is breakdown of geographic barriers. Two, the consequence of crowding of plants, in other words, monoculture in agriculture. And three, the importance of evolved resistance of crop plants to pests. Okay, let's look at another example, wheat rusts caused by various species of Puccinia fungus. First example here is stem rust, Puccinia caused by the fungus Puccinia graminis, and that's pictured in the photo bottom left of screen. Wheat stem rust. This has been a serious disease of wheat from the times of earliest agriculture in the Middle East. And it's been a problem on wheat in Australia since the arrival of the first fleet at Botany Bay and the first attempts at growing wheat in Australia. It's controlled in Australia mainly by growing wheat in drier regions. So that is targeting one vertex of the disease triangle, targeting the environmental factors. Okay, bottom right of screen, we have another, what they call pathotype or strain of rust. This is wheat stripe rust. So rather than primarily affecting the stems, primarily affects the leaves. It's also caused by a species of Puccinia called Puccinia striiformis. But this species of rust has only been in Australia since 1979. Was first found in the eastern states of Australia. The problem with this particular type of rust is that soon after it was first detected, it mutated into several different pathotypes, so you can think of them as strains or races of the same species. And as a result of those mutations, it started infecting more and more varieties of wheat. So big problem. Another example on the next slide, coffee rust. Coffee rust destroyed the entire coffee industry in Ceylon, now known as Sri Lanka, and Java in the 1890s. These were the world centres of coffee production. Coffee originated actually in the Ethiopian highlands of East Africa. So that was the centre of origin. The rust eventually spread from there to all coffee growing centres. The world centre of coffee production then moved to Central and South America, and the planters in Ceylon or Sri Lanka switched to growing tea. So what caused this epidemic of coffee rust? Well, the pathogen eventually caught up with a vulnerable crop. Why was the crop vulnerable? One, because it was a monoculture. Two, the plants had lost their genetic resistance after being cultivated for a long period of time without the pathogen being present. So again, we're reducing the genetic diversity of the plant by moving it to a different biogeographical region in the absence of that selective pressure. In other words, in the absence of the pathogen or the pest. Yes, of course. Where does it come from?
You know what I mean? Like, it's not present.
Yeah, it's not present,
Present.
so it must be transferred there in some way.
See, because I remember like when I was like 10 years ago when I was back home.
Yeah.
So we had like mice and there was this insect.
Mhm.
Never made the problem. We never heard from it.
Mhm.
Then one year, because we have like an antibiotics where I live.
Yeah.
The airplanes, I don't know how. Anyway, it came through maybe, I don't know how. And it decimated.
And suddenly it was just present in huge, and it just
It's a big bug.
Yep. replicated out of control.
Out of control.
Yeah.
So actually somehow the pathogen needs to be transferred.
It has to be transmitted. It doesn't appear out of nowhere. Doesn't appear out of nowhere. It has to be transmitted. And now, as you can imagine, with the way that we move around the world, all the different transport mechanisms that we have, and how quickly we can move, even with the wheat stripe rust arriving in Australia, they don't know how it got here. They believe that it was on someone's clothing. That's all it takes. And because people can move around very, very quickly, spores or other reproductive structures survive over those short periods of time. And then if they take hold in that new, geographical region, then suddenly you have this new pest or new pathogen. Yeah, good point. Okay, our next example is chestnut blight in the USA. This particular disease killed most of the magnificent chestnut trees in the mountain forests of eastern USA. They were killed by a fungus which is transported or transmitted by wood-boring insects and was carried to the USA from related trees in Asia, where it does little damage in Asia, but in the USA, it encountered trees with no resistance. Once again, so you can see these common themes coming through again and again. Another example, Dutch elm disease, also has an insect vector. It's a fungus with an insect vector. The fungus is carried and inserted into tree trunks by a wood-boring insect. Again, spread from Asia, where it occurs naturally with the local elm trees in Asia, and it does very little damage there. But when it was transported accidentally to Europe and then Northern America, it killed just about every elm tree. Again, no resistance to that particular pathogen. It has spread to New Zealand, but not yet to Australia. We already have the insect vector, but not the fungus. It's only a matter of time. This emphasises our biogeographic isolation in Australia and the need for quarantine to exclude pests and diseases of importance in both agriculture and horticulture. Note that in Australia, because of our geographic isolation, quarantine restrictions on importing plants and animals has been crucial in excluding many of these pests and pathogens. of which there are quite a few very, very serious level 2 pests and pathogens. If they were to arrive in Australia, not only agricultural crop plants, but stock animals could potentially be devastated. Another example, eucalypt dieback, caused by the fungus Phytophthora cinnamoni, has killed more than 300,000 hectares of jarrah forest in Western Australia, plus thousands of hectares of native forest in Victoria after it was accidentally introduced from the tropics. It does very little damage in the tropics, but again encountered susceptible plants in southern Australia. And you can see this whole region here in the photo, gone in this photo to the right of screen. Example, black leg of canola caused by Leptosphaeria maculans. This destroyed the budding canola industry in Australia in the 1970s. The then Department of Agriculture, led by Dr. Phil Salisbury, selected and bred resistant cultivars of canola by exposing a wide range of genotypes to the disease in fields in which the pathogen had been allowed to build up in the plant debris. They selected durable resistance, and since then it's been shown that genetic diversity and rotation of different canola genotypes is important in disease management. Because if you simply have one canola genotype with one form of resistance or tolerance, then what happens? The pathogen will circumvent that tolerance or resistance. So it needs to be mixed up all the time. Next example, phylloxera and downy mildew of grape. So here we're looking at an insect parasite and pathogen. Grapevines suffer several destructive pests and diseases. Downy mildew, this is a very common one. Powdery mildew, grey mould, and this insect pest here, phylloxera, is a major one. Downy mildew, a fungal disease, devastated old world domesticated grapes after it was first introduced accidentally to Europe from North America, where it had co-evolved on wild grapes on which it caused little damage. It was introduced when phylloxera-resistant American rootstocks were taken from America to France to try to control the insect pest phylloxera, which had also previously been introduced from North America. So again, all this movement around the globe and all these problems that result. Another problem, in Europe, the grapes were actually grown as a genetically uniform monoculture. That is, they were grown as clones. Yeah, vegetative propagation, clones, and they were highly susceptible types, no genetic diversity. And this is still the case today. Okay. Next slide, let's look at downy mildew, or rather the symptoms of downy mildew. And we can see up here, as indicated by the arrow, what looks like an oily spot on the leaf. Now, this is the earliest sign. It's a single leaf spot every 50 metres or so of vine row. Upper right, they look a little bit more subtle here, but you can still see those oily looking spots. And often these are not detected until the disease spreads and they become more visible. Okay, this image here, now we can clearly see the white fluffy growth, which is the spores, the hyphae and the spores of the downy mildew fungus, and these will develop beneath what appears to be oil spots on the leaves. These develop after warm, humid nights. And then lower right, here we have rapidly growing oil spots, which often merge to infect much of the leaf. And of course, you would see that as necrosis of the leaf at that advanced stage. Okay, let's look at now phylloxera, a destructive insect pest with a typical pest history. Phylloxera is a small aphid-like insect that feeds on roots with a stylet mouthpart. It's a destructive and famous pest of grapevine. It has a long and important history in Australia, and it's actually regarded as the world's worst grapevine pest. It evolved on undomesticated grape species in North America, and then it was introduced with the rootstocks to Europe. And by 1880, it threatened to destroy the French grape industry. First appeared in Australia near Geelong in 1877, and it more or less destroyed the grapevine industry in the Goulburn Valley, the Yarra Valley and Geelong region in the 1890s. But it never quite got to the main grapevine areas of Western Victoria, Barossa Valley and Hunter Valley. It's still present in parts of Victoria near Sydney, but it's restricted. These areas are restricted by quarantine. A more recent wine industry is developed in southeastern Victoria, which are commonly referred to as the old phylloxera districts. And these regions have developed based on the use of phylloxera-resistant rootstocks selected from those American grape species. And this is what phylloxera nymphs on the root of a grapevine look like. Phylloxera is a tiny pinhead-sized yellow wingless aphid-like insect. The fibrous roots that are infected with phylloxera develop fleshy yellow galls, and you can see a photo bottom left of screen. The insect populations are most evident in midsummer to autumn, and they're more common in heavy soils that crack and pull away from the roots. This allows the phylloxera insects to move around. Phylloxera is not so common in sandy soils, which actually don't permit easy insect movement. Okay, symptoms of phylloxera infestation: weak growth of grapevines, reduced cropping, premature yellowing in autumn, decline leading eventually, but over a long period of time, to vine death. And just have a look at that photo bottom right of screen. So what we're seeing here is one of these galls that develops on the root. And if you were to dissect open that gall, which you can do very easily in the lab, then you might see a phylloxera aphid, this circular looking structure here. This is the female. Yeah, it's a type of, it's an aphid-like insect. Yeah. Yeah, no, these are the eggs here. Yeah, they're the eggs. But the female just looks like this spherical structure here. Yeah, that's what you would see inside one of the galls. So you can see many galls here on the left, galls that develop on the roots. Okay, main control of phylloxera is the use of phylloxera-resistant rootstocks. Quarantine is crucial in restricting spread to uninfested areas. The insect itself has a very slow natural spread. They don't tend to move long distances like rabbits and cats do. So long distance spread from one region to another will always be as a result of human transmission of the insect. And you can see the efforts that are still in place there to prevent the spread of phylloxera. Okay, let's move on to our next example. Very important example. This is the cotton boll weevil in the southern USA. And this example encapsulates the history of agricultural entomology, integrated pest management, which we'll come to in a minute, and the evolution and again social and political importance of a crop epidemic. Let's start with the history of the cotton industry in the USA. 1620, cotton from the West Indies was first planted in the colony of Virginia. For example, in the region known as Georgia, this is a coastal southern subtropical state of the USA. In 1733, the colony was founded and cotton was first planted here in this year. In 1743, with the use of slaves, Cotton production expanded rapidly as part of the colonial trading system designed to increase the wealth of the English. Slaves were taken from Western Australia. They were abducted from, Western Africa. What did I say? Western Australia. Slaves were abducted from West Africa and taken to the Americas to work on cotton, sugar, tobacco, and cocoa plantations. Then the raw cotton, sugar, cocoa, and tobacco was exported back to England. Cotton textiles made in England using new mechanical manufacturing techniques, i.e. the Industrial Revolution, and exported from there. By the year 1790, there were 700,000 African slaves in the USA, most of them working on cotton plantations. 1850, by 1850, there were 3 million African slaves in the USA, 2 million of those working on cotton plantations. Cotton was the basic raw material that started the Industrial Revolution in England, in the cotton mills of Manchester, and also contributed to the early wealth of the USA. Cotton was the greatest export earner, paid for 2/3 of the imports into the USA, and supported massive immigration. For example, 6 million people just in this 10-year period from 1850 to 1860. The cotton manufacturing industry in the USA exceeded England's. All slaves and the poorer white population were entirely clothed in cotton. Around this time, Levi Strauss began making their famous jeans. 1861 to 1865 enter the American Civil War and abolition of slavery. The cotton boll weevil was first described in 1843 from cotton in Mexico. And in 1862, it caused abandonment of cotton growing in one part of Mexico. It became such a serious pest. It was first reported in the USA, in Texas, in 1892. The cotton boll weevil was actually introduced accidentally from Central America. By 1903, 50% of the cotton crop had been destroyed by the cotton boll weevil. Attempts at eradication were given up and biological control attempted by introducing predators, for example, a mite from Mexico and Cuba, but this was not successful. I'm not sure of the reasons behind this. Attempts were made to quarantine the weevil in Texas. In 1908, the first edition of the Journal of Economic Entomology made one of the first recommendations of dusting cotton plants with lead arsenate powder. Wow, lead arsenate powder, one of the first insecticides, 1908. You don't have to be a chemist to appreciate the potency and danger of lead and arsenic. 1915, so that was 1908. 1915, the cotton boll weevil then became observed in Georgia. By 1917, cotton production had fallen by 30%. By 1923, output had dropped by 80%. You're starting to appreciate what an enormous problem the cotton boll weevil was. Here it is, Anthinomus grandis, the cotton boll weevil that you can see to the left, top left of screen. It has that characteristic long snout with the antennae attached to the snout. The weevils, or larvae, often feed on seeds, and so they're common pests also of stored grain. 1920, 4 million kilos of calcium arsenate dusts were being used on cotton annually. Farmers were advised to dust every 5 days after flower bud formation. So this was an equivalent of 10 dustings per season. 1920 still, The transition was made from man, mule and horsepower to mechanical power on farms, including the use of aeroplanes to spray the cotton. And you can see that in the photo bottom right of screen. Still, losses of 20% were occurring. This did not include the cost of attempted control of cotton boll weevil. In 1927, the partly resistant Acala cotton was discovered in Mexico, in Acala, Mexico, and then became widely used in Western USA. Note that they were searching for resistant varieties or tolerant varieties to cotton boll weevil in the centre of origin of the crop. That is a Vavilov centre for cotton. 1938 to 1947, the flower buds were being mopped once or twice with a cloth dipped in calcium arsenate syrup of molasses, and then dusted 3 to 4 times with calcium arsenate dust as the cotton bolls were developing. Wow. Don't you read that and think that this is just crazy?
They had to do something.
They had to do something, but really?
Well, at the time the technology was offering that.
Would you do that today? If your crop was seriously affected by a pest or a pathogen and you were losing 80% of your crop, would you turn to arsenic or lead or any such toxic chemical to save your crop?
So you think all the time it was that toxic? That's my question.
Arsenic had been used for poisoning for a long time before that, even to poison other humans.
You know what I mean?
Yeah.
Sometimes it's just that
I know. It's not food. It's not even a food crop. But I know it gives people work and that keeps them alive. Yeah. I don't know. I know it's easy to say these things in hindsight. Absolutely right there. But doesn't it sound crazy after the fact? After the fact, it sounds crazy. You're trying to appreciate why it was done then, why they were turning to such extreme measures to save those cotton crops. The advent of organic insecticides, chlorinated hydrocarbons, CHCs, and then later on the organophosphates, the OPs. After 1947, a wide range of organic pesticides, DDT, toxaphene, chlorinated hydrocarbons, in other words, CHCs for short, developed. But they developed for control of mosquitoes and other vectors of human diseases during the Second World War. They then started to be used against the cotton boll weevil. In the 1950s, more than 30% of all insecticides made were being used on cotton, usually mixtures of 2 insecticides with sulphur dust. But the boll weevil problem was as bad as ever, even after using all these different chemicals. 1950s, they used low volume sprays instead of dusts. As technology was advancing, they also used new, more toxic chlorinated hydrocarbons, such as aldrin and dieldrin, weekly sprayings of these chemicals. 1954, the first report of cotton boll weevil resistance to chlorinated hydrocarbons emerged. They then found that 65% of weevils were showing some level of resistance. So they swapped from CHCs to OPs. Malathion, parathion, very, very toxic parathion. Then what happened? Formerly minor pests of cotton became serious pests as their predators started being killed by all these pesticides. For example, plant bugs, thrips, other bollworms, budworms, and so on. 1968, so not that long ago when you think of it. Well, maybe for you guys, but not for me. 1968, 10 to 14 chemical insecticide treatments by tractors were being used, plus 3 to 4 aerial sprays per season. 1972, Losses reported of $300 million despite 60 million spent on control. Half of all the insecticides produced in the US were used against the cotton boll weevil. 1962. This is an important year. Publication of the book, it's just a small book, Silent Spring by Rachel Carson. Who's read this? Get a copy from the library. We have copies in the library here. This is a must read for any ag student. Okay? Silent Spring, and it's by Rachel Carson.
And when you go inside chemicals, forget about it.
No, not forget about it. Not forget about it. No.
I know what you mean,
Yeah,
but I'm
yeah.
saying, like, she's the one that says, don't spray.
No, she doesn't say don't spray. She says use chemicals responsibly. And she gives so many examples of what we've done. Yeah, and the outcome. Brilliant. Anyway, Rachel Carson published Silent Spring after years of research in 1962. Brilliant book. I have a copy. I've read it several times. I'm still reading it a 10th or 11th time now. A brilliant critique of the problems of uncontrolled pesticide spraying, uncontrolled misuse of chemicals. The book is not saying don't use chemicals. Don't think of it like that. When she released this book, the pesticide industry and lots of different individuals tried viciously to discredit her. They couldn't. There was too much evidence out there. She'd done her research too well. Not just her, all the people that helped her. They had to take notice. Yeah, the public started taking notice. Politicians had to take notice. Yeah. And slowly the agricultural fraternity came to accept the ecological reality of insect damage and misuse of pesticides. Basically, the ideas that you're being taught and will be taught in this subject. In the 1980s, the cotton boll weevil IPM programs started to be developed. Integrated pest management programs were developed. This is just a very brief summary of those management practices that were developed. They returned to the original pre-pesticide methods of early planting of short season varieties to try to avoid the life cycle of the pest. They started to use partially resistant varieties, breed and use partially resistant varieties. They used only moderate amounts of fertiliser and delayed or reduced plant thinning, which was used to stimulate early flowering, again to avoid the pest. They would shred and plough the stalks in winter to reduce overwintering of the weevils. Yeah. So there's one example of where that stubble retention, no-till, it's not going to work. Not in this crop, in this region. Yeah, okay, keep that in mind. They would shred and plough the stalks. So they're getting rid of those overwintering weevils that are going to contribute to the next season epidemic. Pest surveillance started taking place and spot spraying. So they were still spraying, but it was targeted. Spot spraying of hotspots where the weevils were actually developing, and they used organophosphates. Reducing survival of the dormant weevils by late autumn spraying. Again, it was targeted spraying based on surveillance, that is monitoring of the pest, and using less poisonous insecticides, malathion and the pyrethroids. They started trapping emerging weevils in the spring using pheromone traps. The profits from using short season cotton and all these IPM strategies that we've just mentioned were from 50% to 130% greater than using their standard full season chemical pesticide treatments. The proof is there. Proof is there. The number of sprays reduced from 18 in 1971 to only 1 or 2 in 1990. Pest control costs dropped from 26% to 6% of the total costs. So even implementing all those different IPM strategies, the actual control costs were much, much lower. Pest control measures, the rise of the industrial chemistry in 1900s, and especially after 1945, led to dominance of pest control by spray application of toxic chemicals. Use of chemical pesticides has been more extensive for control of insects than for fungi. Why? Because it's easier to develop chemicals that kill insects and not the plants than to develop chemicals that kill fungi and not the plants. Okay? Or perhaps more effort was put into the early development of insecticides because they were actually being used originally to control insect vectors of human diseases such as malaria and typhus. So crop resistance strategies has been used more for control of fungi than insects. The use of crop resistance has been neglected somewhat for control of insects because of that ready availability of chemical insecticides. This has all led to problems of resistance to pesticides, killing of beneficial organisms, especially parasites and predators of insects, thus creating new pests, human toxicity, and of course, widespread environmental pollution. Now attention is returning to more ecologically sound and sustainable methods of pest control using crop management options, that is looking at planting dates, intercrop treatments, cropping periods, plant spacing, mixed cropping, crop deployment in paddocks, control of irrigation and crop rotations. In other words, all those different economics of farming. Crop plant resistance, still an important strategy. Selective use of chemicals to avoid harming beneficial organisms and developing pest resistance to the chemical. And finally, biological control, that is promoting the beneficial organisms that control the pest organisms in the first place. Okay, we've looked at the disease triangle. We can also consider this as the damage triangle. It's useful to discuss the cause of damage or disease as a triangle of interactions, as we've seen, between a host, plant host, the pathogen or pest, and the environment. There are many interactions that are at play here. And this can be initially developed for each pest or pathogen and then assembled into a whole crop picture. Control measures can be targeted at all corners of the triangle. And a final note on the ecology of human disease. There are actually some similarities between the ecology of human diseases and crop pests and diseases. Most crop and human diseases are crowd diseases. They initially develop and are favoured by crowding together of humans or crowding together of plant hosts. Most human diseases originated in crowded cities and were non-existent in small nomadic populations. These small populations would not be able to sustain a serious pathogen. Therefore, small populations tended not to have resistance to diseases. And they were actually badly affected when the pathogens were introduced during colonial times. So, for example, Aboriginal peoples of Australia and America had few pathogens before colonisation and were devastated by introduced pathogens. The big difference is that human populations remain genetically outbreeding. and we retain genetic diversity as a basis for natural selection for disease resistance. That is, we remain genetically diverse and therefore genetically adaptable. Plants do the same thing, but we don't allow them to do that the way that we manage them.